waiting for the possibility to have a live event here in Genoa, we decided to start with this initiative to put together the number of talks under the form of webinar. So this is an experiment. So I apologize if something might not be going perfectly smooth. I try to manage everything in the best way as possible. So the nice thing of the experiments is that we must be, and we are flexible. So we recently added on the web page of Protelec Electrostatics leave a feedback tab, so you can put your feedback and give us your opinion and suggestions to improve the organization of the webinars. You are very welcome to do so. As you have been seeing in the emails and on the website, the way to attend to the meeting are two. You can either register and get the Zoom link, and then you can actively participate to the meeting and to the discussion, which will take place afterwards. Or you can use the YouTube link, which is on the web page, and then you can ask questions only through the chat on the YouTube channel. And in order to do this, you need to be authenticated on YouTube. After the presentation, after a few days, the presentation will be put and will be recorded and put on the YouTube channel. I don't know for how long they can be on the web because of the privacy law. I think at least 1 year, but I don't know if they can be there for longer than this period. Okay, so I would like to thank Pedro Raiz from Lisbon, who helped me setting up the web page and to organize the webinars, and Panagiotta, who is our secretary assistant for other bureaucratic issues. I would like to add that so one of the reasons for this webinar series is to try to involve youngest researchers also in this period of lockdown and pandemic. In order to do this and to motivate them to present, we added the possibility to submit an answer for flash talks. It will be 10 minutes long talks that can be put after the full-time presentations. So I would be pleased if the youngest among us want to try this possibility. And there will be plenty of space for discussions. We will see how to organize this as long as the series goes on. As I told you, this is an experiment. I will try to do my best to provide the support for all the possible connections among us and the presentation also of the students. So then I think Antonio Batista would like to add, which is one of the organizers, together with Marilyn Brunner and Bertrand Garcia Moreno of the Protelec Electrostatics. So maybe Antonio, you want to say something?
Okay, hello everyone. Good afternoon, good morning, good evening, whatever it is for you. Well, it's great to see this webinar series moving on. This whole thing started some years ago in 2014, I think, when I was challenged by Marilyn and Bertrand to do something in Europe similar to the Telluride meetings on protein electrostatics in Colorado. And then after that, Walter Knapp and then Anna Demjanovic, and now Walter, they kept this moving on. And I especially need to thank a lot to Walter, because I mean, in this complicated situation of the pandemics, he had the idea of this webinar series to keep this alive and kicking. So it's, thank you, Walter, thank you very much. For now, it's still a virtual kicking, but soon, hopefully, it will be the real thing. Although keeping these webinars going on as well, it's great, and I think it's great that you mentioned the younger people to be strongly involved in this, because this meeting is in a large part of it is designed for them actually. So thank you, Walter. Thank you so much, and thank you for the people that were previously involved in this. So yeah, I think that's it. Thank you.
Okay, thank you, Antonio. So I would like just to add that we recently got into the CCAM events from the north. So I think this event will have even more visibility, especially in the European scientific community. So having said this, I think it is time to start with our speaker of today, who is Jim Warwicker. Jim is a physicist by background. He got a PhD in biochemistry from Bristol under the supervision of Herman Watson. And then he spent 5 years at Yale in the group of Fred Richards and Tom Stites. And then he came back to the UK after working in a UK Research Institute. He moved to Manchester in 2000, where he is been since. He will be talking today about the spike protein, which is a very hot topic in these days. So I'm eager to hear from his presentation. Thank you, Jim, for being here.
Thank you very much, Walter. Oh, please let me know if I'm mumbling too much, or if my connection goes down, or if you can't see things. I'm assuming Walter, can you just tell me you can see things okay right now?
Yes, we can.
So thank you. So thank you again, Walter. It's brilliant that you've put the effort in to get this going again, and obviously there's a lot of interest. I don't think necessarily in my talk, but just clearly in the community. And I'm not looking forward so much to my talk, but I'm really looking forward to other people's talks. So I just would emphasize again what you said, that people can contribute and keep it going. So thank you. So my talk will be quite short. As you say, I really welcome discussion at the end about what people think in general about our response to the pandemic. I mean, clearly this angle is just one angle purely from my background, and other people will be thinking along similar lines or other lines, and it would be great to hear people discuss that, and there should be plenty of time for that. So because I have been around a while, I can put in papers from the late 80s and the early 90s, which is when I was thinking about these were pico-RNA virus capsids. That's because Michael Rossman, Jim Hogel, and then later David Stewart, well, not much later, the foot and mouth disease virus. It was rhinovirus for Rossman and poliovirus for Jim Hogel. And so, you know, I don't know, we didn't call ourselves bioinformaticians, but as a computational biologist, it was interesting to look at those, especially in regard to the pH changes. So these pico-RNA viruses were classified originally, as far as I can recall and understand, according to their response to pH, just biophysically, but then in vivo, a rhinovirus wouldn't pass through. I've forgotten the details, but poliovirus would be, I believe, would be stable to stomach pH, whereas rhinovirus wouldn't, and so on. And foot and mouth also was quite acid labile. So there's there's a degree of pH dependence there, and obviously the atomic structures becoming available from the great work by the Rossmann et al. Labs stimulated that thinking. This work, that the 12 and the 29, I don't know if you've worked out what the 12 and the 29 on this slide mean. Probably have. They show the great degree to which the world took up the work that I did at the time, the 12 citations for one paper and 29 for another. So it didn't go that far really, although other people have been looking at things, related things, and doing more work subsequently. One of the things in the 80s, actually, I remember when we're lucky enough to be in the big labs have got visitors, and every lab's got visitors, but big labs tend to have more visitors. And one of our visitors was Don Wiley, and I remember him discussing. I guess he was visiting Stites and colleagues, Tom Stites and colleagues. I remember him discussing. I was quite dejected actually with this study that they published in discussion of the fact that if you look at the interface in the trimer of hemagglutinin, and I've already forgotten whether it's a trimer or a dimer. That's dreadful. That's in hemagglutinin interface, and you look experimentally at which of the mutations that cause a shift in the pH of fusion. And I think, I don't know, maybe I'm alone in this, but I think as computational biologists looking at proteins or other molecules, we're looking for single regions, single groups. That's the switch that does it. And of course, it was all over the place. It was all through the interface. And I presume the obvious explanation for that is if you've got two or more conformations and you alter the energetics of those conformations in any way, then you alter the balance, and therefore, in this case, you're shifting the pH as observed. I'm not using my pointer, the pH as observed here. But of course, beyond that, there still has to be groups that are giving you a pH dependence, which are changing their charge which are giving you a charge difference with respect to difference between the two different states, and therefore are giving you a pH dependence. So we came up with this obscure idea, which I think is obvious to those in the community, but I tried to draw it in the most obscure way possible, working with Eisen Satanabats. We were working on acid sensing ion channels, and I look at this and I just I despair again, I'm afraid, looking at what I drew here. This wasn't Ivan's doing, he did the proper work. I tried to summarize the idea that you've got everything can be pH coupled if it's affecting interfacial energetics, whereas you've got a few groups that are going to actually alter the pH dependence of the energy in terms of the slope. And if you alter if you've got a slope, you can move it up and down and you can get different pH, but you want to therefore distinguish between groups which are pH sensing, which we're really interested in, and the pH coupled ones, which are just changing the energetic conformations. So part of the problem maybe in computational biology is to draw things better, which I think normally means we understand things better because we can put them down more simply. And I didn't do a good job on that one, and I still don't understand it perfectly well. But there are I noticed people here who will know much more about these things than me, but they will also know, they'll even know that my contribution to looking at this was really to use the methods of other people, starting with Don Bashford and Martin Karpus and this paper as well, for that general idea of how to set up PK calculations, and this paper on how to do Monte Carlo simulations to get the PK values, Monte Carlo simulations of the protonation states. And then I think it's 2004 publication was sort of summary of how I'm doing that. I'm always lazy in trying to think about how to do things. And so my idea to try and avoid, in the most simplistic way possible, doing large-scale conformational searching, which is clear to me now is one of two of the major blocks on pieces of work now for thinking about any spike sort of problems like that. And I'm sure it's evidence to many people here, is that number one, we need experimental data. If we have a suggestion, we need experimental data to check it out. Number two, we do need to look at different conformations, and whether or not you do that computationally and or experimentally. We're often talking about, well, what happens in different conformations, or if we change residue, how does that change the conformation? And therefore there's people much more expert than me at those sorts of methods. So the idea was to find a finite difference plus Boltzmann method, FDPB, but we're also concerned that we're locking ionizable residues into conformations they don't want to be in when pH changes, when they titrate or ionize or deionize, and therefore I linked it with a Debye-Huckel simple water everywhere method, so that we could always retrieve if a distinguishing between residues that are buried and they're stuck in finite difference plus and Boltzmann, than ones which are less buried and can always get to water dominated, not so interesting sort of interactions. And then actually Walter described that he has a link to people to help him a little bit with the web, although obviously he put a lot of effort into this, and I am bound by my own inadequacies in many ways. And whereas Max Hebditch is young, who's this person here who worked with us for several years now, has left to a software engineering job, and software engineering was one of his strengths, was the thing that he really wanted to push, and therefore, and I'm sure many people do this, but Max is a particularly good example of someone really competent in being able to take tools and put them on the web in a very simple way. And so we've got this server protein sol, which originally was designed to look at sequence-based protein solubility prediction, largely because we were getting money and have good collaborations with good colleagues looking at solubility of antibody therapeutics and protein therapeutics in general with industrial collaboration. So we had that, but then we've added other tools, which I will just briefly show before I get on to what we are going to talk about. So this is Anthony Nichols reinvigorated after just showing what Anthony Nichols showed 30 years ago with graphs. We can do patches too, like a Pymol-Can, a SysPP viewer, and so on. But we're also interested in trying to say, well, we think this is sort of hydrophobic area here, but what about if we specifically color it in terms of its ratio of surface area of polar to non-polar groups? And so we've got that going alongside too. And then we use that to compare antibodies and so on in other systems and try and say, well, this is charged, this is non-polar, that might interact, especially around the CDR region. So we call that our patches option. We use the simple Debye-Huckel method just to quickly scan through predicted pH dependence for input structure files over pH dynamic strength. And we can do all these calculations because it's the very simple method, again with the Monte Carlo sampling of protonation states. And so it shows us things you know. I mean, it gets more charged if you go to more acidic pH, which isn't that great a surprise, really. But what was nice was that we found that these are domains of antibodies. We found that the partitioning that experimentalists got for these domains, which is in the black lines here, seemed to correlate with what we get. So though we're doing the obvious things about charge changing and energy changing from more stable to less stable as you get away from neutral pH normally, there was some indication of correlated with experimental measurements, and so we're pleased about that. And then this was something that Max Hebditch did himself. He wanted to introduce some machine learning models to try and correlate with or make predictions for monoclonal antibody biophysics, because there was a big paper about 3 or 4 years ago now with those biophysical properties, 13 different biophysical properties, ELISA against a bunch of other proteins, and then HIC is the ones easy to recognize, hydrophobic interaction chromatography. So these different measurements that people might make for antibodies to say how developable they are. That's a real word, developable, something like that. I think it is, for the bioprocessing community. And this doesn't look like a brilliant correlation between predicted and experimental. And this is for HIC, hydrophobic interaction chromatography, but this is about as good as it gets. And it certainly couldn't. Max spent a good deal of effort looking through machine learning models to get this out. So about the same number of slides now for what I said I was going to talk about, and that was there looking at another tool that we have on the server, which we put up in March, because I have this FDDH code, define difference plus and Boltzmann, whatever code, and that takes about, for a trimer of spike, so what's that, about 25,000 atoms, a trimer of spike proteins. It takes about up to 24 hours on my system to run. And that wasn't really sufficient to get people to look at it. And so I did the obvious thing, which I just put off doing for years. So I even simplified it further by saying you can look at where you think residues that might be interesting, where they might be buried. So give you a better plot of burial. And then you say, right, I want to center a calculation on there. It does a calculation around a certain radius and gives you those results. And then you can go to other regions and get those results, and they accumulate. And this is one where I've done all of them, obviously, sort of offline, but the online tool does different regions and then aggregates them together as we go through. So people can use that tool should they wish to look at pKa predictions. And then obviously we can use it offline to look at bigger systems. And I did do, when we published this in Bioinformatics this year, we did this because it was locked down and we wanted to do something. Max actually was just leaving, and I thought I would try and get him to do this before he left, to put the stuff on the web, which he was very, very helpful with. And this is a benchmark, one of two benchmarking things, and this was against David Baker design helical protein, where they put histidine in, which is, I recall, ionizes and separates the multimer, and the histidine sticks out in these calculations. Benchmarking might be too strong a term. It's consistent. The calculations are consistent. What's known experimentally, and obviously it's not a blind test, not a proper test. And secondly was hemagglutinin. So if you look at hemagglutinin, and you have a whole bunch of residues which are more green here. So more green is, we think, is more buried. And that's a simple solving accessibility calculation. Going back to Fred Richards and BK Lee. But not all of the green ones are frustrated. So what we're trying to look at now is blue versus red. Blue is happier, and the more blue it's, the more energetically stabilized, the more stabilized in the delta pKa, and the more destabilized in the delta pKa, the more red it is. And so it doesn't, if they're buried, they don't have to be destabilized, and they can, so we know these things, but we can see these. And then we try and match those to what my impression of what literature says about what groups are involved in which regions of hemagglutinin. And it seems to correlate. So again, benchmark might be a bit strong, but it seems the residues we predicted should be it should be destabilizing at neutral pH, and therefore might contribute to a pH-dependent conformational change in hemagglutinin. Seem to correlate with what they do correlate what's seen in literature over many years of study. So again, I would invite people to talk about this afterwards if people have ideas about why they're in, what work they've thought about doing or might do. So I think the thing I want to say here about coronavirus, everything is my impression, because I am not a cell biologist, and we can all put up these slides, but we can do them in my case with very little knowledge. What's relevant for me here is that the virus either fuses at the plasma membrane and releases its genome there, or it gets endocytosed and goes through endosomes. And I don't know what the current thinking really is of what the predominant route. I think the current thinking is it can do either route, and coronaviruses in general can do either route, and some coronaviruses do more of one and more of the other, plasma membrane versus endosome. But it seems that it depends on the protease sensitivity of the S1, S2 cleavage in spike, and on what proteases do that, and what proteases are available either in the endosome or the plasma membrane to do that. And I think people are leaning towards that if everything is in order, then maybe it's more, I'm not sure, but maybe it's more by the plasma membrane, I don't know. But then of course you've got everything that's pertinent to what I'm talking about here today. But of course there's everything else, and what people, with this slide from this paper, what people have been thinking about in terms of therapists. And I must admit, so we're allowed, because it's COVID, we're allowed to sort of just say our complete layperson comments, which is, remdesivir is really disappointing. Again, people that know more about this, please say something, because one would have thought it was developed, wasn't it, for other viruses, for Ebola and other viruses that use RNA-dependent RNA polymerases, RDRPs, and mechanistically you'd think, well, it's going for an activity that's uncommon in humans, and if you can get that activity right, you should be able to take out the viral pathway. So again, I don't know detail of what's gone wrong with those inhibitors, why they're not working effectively enough. I mean, and there will be information on that, of course. Right. Oh, and this is an even more layperson comment. And when you look, so the first thing you think about, a bit like hemagglutinin in an influenza story, is well, there's pH dependence. If it is going through the endosome and there's acidification, and also you're looking at the fusion process. So you're looking at how the spike binds to the receptor. S1 is shed, fusion peptide comes out and hits the opposing cell membrane. And then that's sort of where it tends to stop crystallographically. I mean, in some indication, the fusion peptide is coming out in just a few, sorry, I say crystallographically. Structurally, of course, that's all changed with cryo-EM. But still, it stops about here, and you don't tend to think about the horrific visualization. I mean, that actually these two are supposed to fold back again. The heptide repeat parts are meant to fold back again, and to bring the membranes together, and then to bring the membranes together on two different sides. And so you make a channel, as I understand it, through which the RNA can depart the virus and into the enveloped virus and into the cytoplasm, either via the endosome or via the plasma. So again, there's cell biology there that one has to try and take more time than I've used to understand. And this is actually, I'm not showing, I don't think anywhere I'm showing a proper spike protein trimer picture, which we're all familiar with from our newspaper covers and so on. But this is the so-called post-fusion structure. Although again, given the previous diagram, it's sort of on the way to post-fusion, but the fusion peptide is pushed out, meant to be pushed out up here somewhere for the opposing membrane. So S1 is lost from the structures, it's S2. But when we published putting our pKa predictions on the server, we used this because there was some, I can't really recall why I used this, but I think it's because there was some data that these two mutations, these two glutamines, two histidines, gave rise to pH dependence. And this is for mouse hepatitis virus, coronavirus, but not SARS-CoV-2, MHV, and also the structure for this, for the so-called post-fusion form. And again, I haven't shown it in the pre-fusion form. In the normal spike protein S1, S2 trimer, these glutamines are buried, so the histidines would be buried. So the idea is they would be destabilized, but now they're exposed here, and you can't see any red. So that would be favorable for pH-dependent conformational change from the one I'm not showing, which would be destabilized to this one. And this is actually the cyan here is what the conformation of this part would be in the S1, S2 trimer, and then it's extended in this version here, and then the what would have been buried is 1015, 1042 come out. And what we did see was, what we did think in terms of what would destabilize were these two histidines, but they are in a part of the structure which is conserved. One of the very few parts of the structure that's conserved between the so-called pre-fusion and post-fusion structures, which is I apologize for the inverted the quotation marks I use here, because I'm not sure that pre and post is entirely correct usage of term. But so that's a bit mystifying. It's great to see these histidines, which are nicely destabilized, but they're nicely destabilized according to these calculations, and I think most people would say, looking at them, in both pre and post fusion structures. And so we suggested maybe we tried to grab what was available to us. The only thing left available to us was that maybe there's an intermediate in the conformational change between the two, between pre and post fusion, where they become more accessible and therefore stabilized, and that might bring down some intermediate. So that's a pure speculation based on, we don't have anywhere else to go with them. Yes, they're just a structural feature which can deal with being destabilized electrostatically because of some packing phenomenon or something else. And then subsequent to that work, after Max kindly left me to go to his software engineering job for the UK government, although he's still involved in this work actually, in a minor way by making sure that our website keeps going. That we looked at, because there was an increasing number of cryo-EM structures, and of course we don't always rely on our calculations, especially simple ones like the ones I use. Maybe if we did the calculations for a whole bunch of structures, we would see something consistent. And so this is a whole bunch of spike trimers going this way. So there's three rows for each trimer, each of the monomers, and then this is the ionizable groups in sequence. I've only listed the ionizable groups here. There's no gaps for the non-ionizable groups are left out. And then this is conservation, so darker purple is more conserved across representatives of the whole coronavirus family. I mean, later, not much later, there's not much to go, I'll be talking about human coronaviruses, but this is conservation over all coronaviruses. And then this is there is now, I showed you a post-fusion structure for MHV after, because sometimes on keenly awaiting papers to arrive or structures to arrive, you see the information on in bioRxiv. And one of them that I keenly awaited through the summer of this year was from Bing Chen's group, somewhere in Boston, of the post-fusion structure for SARS-CoV-2, and that's the one here. And here we see the two histidines we'd already located, and they are destabilizing according to our calculations in pre and post-fusion. And there's another histidine which I've missed, which isn't destabilizing in post, but is in pre. So maybe, just maybe, that one does contribute to straightforward destabilize the pre-fusion and stabilize the post-fusion in a pH-dependent way. Other things, it was hard to really make out precisely what was going. I was looking for consistency, and was looking for visual things, and some things were a bit visual and some things not. And I'm going to mention those in a while. Did the same for that was S2. Did the same for S1. So there's no post-fusion here because it's just S1 which is shed. This is the N-terminal domain. This is the receptor binding domain, the S2 binding domain. And again, there's some groups which I've noted down here, which of course none of us can read, certainly not on the small screen I'm looking at. I did group them according to open, up. People tend to call them up or open with RBD conformations, or closed or down RBD conformations here. So mostly they're closed. And then I've tried to note, sorry, I know it's lazy. I've taken the text from bioRxiv or from the review actually of the journal it went to. The review copy, and some of the things we saw. So you might wonder, well, you might think that's all rubbish, or you might wonder it's rubbish, but what did you see? And some of the things we saw a couple of residues which probably other people have commented on, which are apparently helical caps, which is always nice to see. They are real. If you talk to my colleague Andrew Doig at Manchester, who's worked a lot on alpha helix stabilization, B985 and B294. And then you see, we're looking for destabilization and pH dependence, but also we see stabilized salt bridges. And these are some examples here. And amongst those, we've got, this one is close to the ACE2 binding site within S1. This one is at a monomer, I can't say monomer interface within, I guess, I'm trying to work out, I wrote it down yesterday, I keep on writing things down, where the, close to the S1, S2 cleavage site, the first S1, S2 cleavage site, not the S1, S2. I think this is actually an S2, but probably in between the S1, S2, S2 prime cleavage, but I can't be sure about that. And then lastly, and I really will just emphasize this without trying to obscure it, the last comment here is that D614, one would hope that D614 would go boom, but it didn't. It just seems to be sort of a weakly interacting thing without any close neighbors that are consistent. There is a K854, but, and I'm not showing you pictures of this, but for me, this is really interesting looking at it, which is that more recently, there have been, starting I think with a structure from Bristol in the UK, there have been LA is linoleic acid bound structures that bind to a pocket, which I'm going to actually indicate to you. And everything changes with 614 and actually various regions of the interface. The interface becomes a bit tighter. So though we've got lots of closed conformers, and they are I think most people now believe without linoleic acid, most of those structures, you get something which is a little bit more tightly closed in many parts of the interface when linoleic acid binds. And that's certainly true of the region around D614. And at that point, D614 pops up and becomes more interesting there. And at least one group has, well, more than one group has put forward the hypothesis that there's a sort of a latch that 614 is involved with, that's obviously lost with 614G. And then that is now believed, I think, quite well established that 614G mutation leads to an enrichment, a biasing towards the open conformation relative to the D614. So you lose the latch and it becomes more open. That's the idea. From what we see, it seems a latch, it's not clear that there's always one residue that's interacting with. I'm not sure it's even a salt bridge. It might even be a hydrogen bond interaction with a main chain peptide group. So going back to what we did see from, if we did average those columns in those heat maps that I showed you, and then we looked at them for the open, which are far fewer conformations than the closed, and tried to look at differences. So it's difficult to see what I'm saying here. First of all, we get the red regions for the histidines. These are the histidines I saw 10 minutes ago, and these are this is the extra one I mentioned. We think these are interesting. And then there were two salt bridges that I thought were particularly interesting because they appear to be, depending on the degree of closure of the closed conformation versus the open, they appear to be mostly destabilized. Actually, they seem to be more or less, or certainly the aspartate components. So this is D398R355 as a salt bridge, and this is D290R273. But the aspartate is really buried in both, and then the arginine is buried next to the aspartate, but it forms a variable interface depending on what's going on with linoleic acid binding, for example. So it appears that the LA binding stabilizes, pushes the conformation together and actually stabilizes the interaction between 398 and 355, the salt bridge, and according to our predictions, stabilizes the pKa of 398. So we think the pKa here is, the delta pKa can be destabilizing, but less so when it's more closed, and it's more closed when linoleic acid binds. So we speculate that might be coupled with the LA binding and the degree of closure, and perhaps also with the D614. But it's actually quite easy to believe for this salt bridge 398-355 that it is coupled to the LA binding. It's in this region here, and underneath it is the LA binding pocket, and it's in X-ray. So you can look at, there are X-ray crystal structures. Of course, what I've been talking about is cryo-EM spike, which has been fantastic. It's obviously revolutionary for sort of structural cell biology, cryo-EM. And if one was younger, one would definitely think about into that area, I think, or interpreting it. But there are X-ray structures for the receptor binding domain, so one can look at thermal factors, and the thermal factors tend to be higher according to this color coding when LA is not. Same with LA is not bound in here. It would bind in here, but it doesn't here. And so presumably these are more flexible pockets, and presumably LA changes that, and it also changes the spike monomer interactions. And this is the salt bridge I'm talking about, 398-355 is right here. So it's easy to see that they would be coupled. 614 is further away, and yet there is a conformational result around 614, as I've mentioned, that is concomitant which occurs for the structures that are LA bound. It becomes more tightly. So there seems to be a whole bunch of things going on, which immediately gives you the impression that we need to know more about the dynamics of what's going on and understand better about coupling between conformational regions. So just taking this salt bridge, which I've chosen because of its stabilizing versus destabilizing properties. So here it's actually the arginine, the delta pKa, SARS-CoV-2 is stabilized, whereas the delta pKa for 398 is destabilizing. You see that too for SARS, and you see that too for MERS. But you don't see that for two other human coronaviruses now, which structure is available for the, I've forgotten whether that's cryo-EM. I think that must be X-ray structures of the RBD. And so the less interesting here, less energetic stuff going on here, according to the predictions here. And if we now, these are a sequence alignment, so you probably can't see that, so I've blown up the relevant part here. And this is for 7 human coronaviruses, these 5 and 2 others, which is not structures. These are coronaviruses that infect humans. And so these are the ones that we know are nastier than these 2 and the other 2, the other 4, these 3. And so if we look at these 3 are the top 3 in this alignment, and 355 is here. So there's 5 with arginine, and there's 5 with aspartate. So there's 5 with which of these 5 with the salt bridges. 2 of them don't appear to be energetically variable. Presumably there's more exposure and there's not as much going on. So I actually discuss it in the manuscript. There are residue changes, which I can't recall now, around that region, which lead to more exposure. And then 2 of them haven't got that salt bridge. And so they're not particularly interesting. So this salt bridge seems to be more energetically interesting in, how can I say this? This is a very weak statement, in the viruses which are more problematic for humans. So one can only say that one can say nothing more than that. There's nothing one can deem as being causal, of course. And I have to thank Max for all the work he's done. Lorena Zuzic is a PhD student with myself and Pete Bond, who is working on the dynamics of other viruses and also of SARS-CoV-2. And so we have great chats together about this. And then other people have been involved with PhD student Sonia as a PhD student, another PhD student in Singapore, this time with Chandra Verma, a joint scheme between Manchester and Singapore. Colleagues in Manchester, Robin Curtis in particular, and PhD students in his group with whom I collaborate on more on the bioprocessing stuff, but they are our sort of a backup. We need backups for our Friday group meetings and so on, especially in Zoom time. So I've left you with not very conclusive talk, and you might all have gone. I'll see in a moment, but what I would say is that I really come back to the fact that people need more experimental data about different conformations. I have mailed a couple of structure groups to see if they'd be interested in mutating a particular salt bridge and see what happens. But they've not replied to me yet. But if we can get experimental groups to make mutations when we've got suggestions, it's the experiment and also more information about the dynamics is what we need for this sort of thing. But in general, of course, we need to work out where we can fit in the whole process. So I'm afraid that's a rather poor ending. And now I'll see. You're still there. That's good.
Thank you, Jean. Thank you very much for your presentation. Enjoy it very much. So I would, okay, I see that we already have a question. So Marilyn, please go ahead.
All right. Very nice, Jim. And actually, Walter, I really want to thank you for getting this started. I hope we keep going with it. So I have two, the informatics part of this is quite interesting. And so I'm curious whether you have some important arginines. And one of the things that Diane Barber taught us at the Telluride meetings was that arginine histidine swaps are something that show up a lot in cancer because they get away from the pH dependence. So that one thing. And so, and then the second question, I guess, is whether the number of salt bridges kind of help. If I think about them as a spring, and we're going between two conformations, one where they're buried, and so we're putting in spring potential of this, wanting to open up and put them in solution. Do you see any kind of maybe, like energy difference between the open and closed form based on the number of salt bridges?
Hey, thanks, Marilyn. It's great to see you and colleagues. Yeah. So the first one, arginine histidine, I'm aware of that. I don't know specifically here. I am very much aware of the arginine histidine changes that sort of Diane Barber has absolutely pioneered. And I think she's one of the people who I have no hesitation in saying I would really want to copy, not entirely copy, use their work to look at those things too. So I'm interested in that as well, actually. but I don't know if it's relevant. And also, I'm also wondering about the Argentinian. I think there's a sort of a codon. I've forgotten now my codons, but I think there's a codon thing there too. But certainly I know that they are massively overrepresented in cancer. You can see that from. So, for example, for anybody interested in that, just to mention this, you can go to a database like the Cosmic database, and you can get a text file of all the mutations, and you can look yourself at enrichment. And then you have to start wondering about codon changes, and it gets much more complicated. But you can start, it's a beginner's sort of thing there, and you can get involved in that field actually. But I don't know about the answer to that first question here. I don't know whether Argentine is so relevant here. The second question is the way I think about it too for the open and close. So I probably didn't say it. And maybe if I didn't say it, it's perhaps because I'm not entirely convinced of how precise our calculations are. But what we see is that we think, and the reason I'm talked about it in dependence, we think the more closed it is, the more stable, in particular that the aspartate member of the salt bridge becomes, and the more open, the less stable. And so I see it just the same way as you do, that there's a potential that would be pushing it towards closed. And the thing is, I have a bit of a conflict in my mind because if we go from a really open to a really closed conformation, then I think most of us would think, well, most of the action is actually going to be only when it's quite local, when we're really excluding solvent, and we're not really getting, when we're 2 or 3 solvent widths, we're not going to get so much. So I think most of what's happening is local. And so I'm a bit confused. I can see the difference between sort of a closed and a very tightly closed. We really are changing the solvent, but I'm not quite so sure about what's happening when you're moving further away. So I don't know about the conformational transitions between properly open, but certainly that's the way I'm thinking about it, with you, which is why the people who some of them I'm sure are here think about constant pH molecular dynamics to try and look at what's going on some trajectory of.
I don't know. Is there any kind of information about infectivity versus sequence, or just even, that would be interesting to see whether it correlates with anything. Thank you.
Yeah, I don't know. Another thing I find confusing is why people. I mean, I'm just welcome people's comments. We can go to the layperson sort of thing. So D614G, in Europe and particularly the US in general, sort of came to the fore. And if you look at the timelines of the way D614G came in against D614, it came in within about 4 weeks, I think, and it went from like low preponderance to high preponderance, which is very similar to the so-called new variant in the UK, which has got a bunch of mutations, which is coming in, and which is leading to a scare about transmissibility. But people aren't talking about 614 in the same way they're talking about new variant, even though it did the same sort of enrichment thing, and therefore presumably was more transmissible in some way. So I just don't think people really understand now. I think people are looking at it from epidemiology, and because there's such sequencing power now, without being able to know the cell biology, because it's not enough time to sort it out. But it is fascinating, and for the new variant, it's got all these different bits and pieces. It might bind stronger to the ACE2. It might be more immunologically unsurveilled because it's got, I forgot whether it's because it's got bigger loops, shorter loops. And then we've got other bits too. So it's a fascinating area, but the experimental data aren't there.
Do you think that present state molecular dynamics simulation can observe such big biologically relevant conformational changes? We have seen a lot of huge amount of computational effort devoted to the spike description.
I'm not an expert, so what I would say is I think we need a simple way of understanding how. So you've got, like, I don't know, 3 or 4 domains in spike, and then you've got 12, 9 or 12 domains because they're multiplied around, and somehow they're changing, and it's not just RBD going up, there's some other changes further down. And I would imagine that even looking at the sort of network model servers might, just to get an idea of that. So, but I would really welcome comments from people that understand that better than me, because people must be thinking about this. And anybody on the, on the call or whatever. I will say, if we have to try and understand it, because I mean, it's a challenge. People have got to try, I think.
So if anybody wants to make a question, please raise his hand.
Yeah, no, I just wanted to comment. Yes, we are looking at that. So we're seeing some changes in the RBD, but it's a huge protein, right? So you don't see a lot in MD simulations.
How difficult would you Hey, Anna, good to see you.
Yes.
How difficult would you rate it as a problem? I just want to, without telling us what your results are, but how would you, on a scale of problems, would you ever choose this problem, or
Well, it's definitely a difficult problem.
So let me add a different dimension to this difficulty. How would you comment on the presence and the role of glycans on the system?
Yeah, that's a great. I mean, if I was being honest, I would just be quiet and say nothing because I don't know. I think, the glycans change as well. People have looked at this, and people have characterized quite early on. People have characterized it, and I'm aware they've characterized it, and I'm aware that structures have come out with the glycans on. I'm aware that people talk a lot about glycan recognition on both sides, on the cellular side and on the viral side, but I actually don't know anything about it. I'm aware that it's a huge issue for recognition as well as stability. I don't know if anybody else knows.
I think that from the computational side, we try to stay away as possible from that. I'm not sure, but this is now it's becoming very, very interesting.
I think what I've not seen it yet with anybody. What we're waiting for is, what sort of thing Anna was just talking about as well. I think people. I've worked on acid sensing. We've all probably seen biological systems where structural biologists say, think about this as a horse stirrup change or a pump change or something. This is the sort of change you've got to think about. But the mechanism is happening here, and they draw complicated pictures. I've seen it for ion sensing channels, and we're waiting. Those are the sorts of pictures we need. And within those pictures, and I think sort of thing Anna was talking about, and within those pictures, which often comes from structural biology originally, whether or not the glycans are just soft and they just let it happen, and they do, they help against immune surveillance or whatever and stability, but whether or not they actually are blocking and biasing against different conformations, I don't know, and that would be interesting. Obviously, I suppose HDX type. I don't know if people must be doing it, but I've not seen anything yet, I don't think, on HDX of spike proteins. Has anybody seen?
No.
Are there any more questions or comments? So I can say there are now 51 participants to the meeting, so I guess okay. So I see Antonio. Go ahead.
So Walter, I've got a question. So have you got the energy to keep this going? I think this is fantastic. So you're thinking about every 2 weeks or every? You've got a talk for next week, right?
Yeah. I try to keep going every week as long as we have registrations, at least until the date where the live event was supposed to occur, which would be next June. But we'll see how many people register, and we could suspend and then reactivate when we have more demand. Antonio,
sounds good.
Yeah.
Sorry, I got muted again. Okay. Yeah. Well, first a comment about the molecular dynamics of this huge system. Part of the problem is not just that the protein is so big that in principle you need to have membranes. So I think part of the problem is that these things, it's not the size, because when you have membrane systems, if some of the motions you expect to see involve some kind of relaxation from the membrane, then things will be horribly slow. So I don't know if it's the case with these kind of systems. I didn't study them, but that could be an additional problem. I mean, anyway, I had a question is not directly related to this system, but Jim, before you showed us something about in your introduction, you showed us something about the difference between pH sensing and pH coupling or something like that. I don't think I got the difference in the terminology. Would you be kind to explain again what you mean?
Oh, I could try. I mean, I think what I was trying to say was thank you, Antonio. That's a tough one, because I think what I was trying to say was I made this definition. It's been barely cited. I don't understand it myself, but I'll give it a go. Which is simply really that if you've got two conformations and an interface that changes, then obviously anywhere on the interface influences, or anywhere can bias the conformations allosterically or in interface. But to have a pH dependence, we need to have we need to have groups are pH dependent. And so if you look at the stability, if you look at the pH dependence of stability, and you've got one line like that and one line like that, without any pH, you need to have the slopes being different between the systems for them to cross over. If they're parallel, then you'll always have the same equilibrium between the two different conformations. Okay. And so once you've got different pH, if you've got like a histidine that's doing something in a pH of interest, in your mild acidic pH, like we've seen maybe for these viruses, then you can have a different slope and they cross over, and then at that point you might have equal conformations. But then these same lines, without changing slope, can move along if you just change any old packing, an isoleucine or something. And so you can change the pH at which they cross over by changing the packing or anything else without changing the slopes. So first of all, we've got residues that make the slope, and they're we call pH sensing. I mean, it doesn't matter really what we call them. And then we've got any residue can just shift you up and down. That's what I was trying to say. I don't know if that's any clearer or not.
Okay, but when you talk about stability, you mean the free energy curves, that you had in your plot. That's what you mean, right?
Yes.
Yeah.
Yeah.
Okay, I see. Yeah, I will have to have a look at that. Yeah, that's interesting. Okay, thank you.
But that's the original observation of Don Wilder's group. For fusion. If you look for people that did the experiments, which people are doing for years on cold viruses, on hemagglutinin and so on, flu, you can try and select for viruses that go to lower pH by changing the cell biology, by using the drugs that change the pH of the endosome, for example. And you select for different viruses, you let them mutate, select for them, and you expect to get histidines out, but you don't. You get anything that affects the different conformational energies.
Yeah. Okay. Yeah, thanks.
There is a question from our people that are looking at the YouTube channel. There is a question from Caroline Fitch asking, the LA binding site is reminiscent of Rossmann Canyon hypothesis. Can you summarize what your thoughts are about LA's role?
So what's the second part of the LA hypothesis? And can you just repeat that?
Reminiscent of Rossmann's Canyon hypothesis.
Yeah. Okay. Yeah, I think that's fascinating. I remember when Michael Rossmann. I remember when Michael Rossmann who was a fantastic scientist, was really excited about the Canyon hypothesis, and I don't know what happened with all those those compounds were called WIN compounds, the first ones that they put into the pocket, the so-called pocket factor in the canyon. And I asked this of the Bristol, of Christine Schnapitzel, can't say her name properly, who was presenting the Bristol LA structure. I don't remember if I asked it. I think I did. And that was, what's the comparison? Maybe I asked someone else actually. And I think they are thinking about it in those terms. I get the impression that the structural biologists that delivered the LA structure originally are working with computational biologists, Adrian Mulholland, I think, to think about what to put in that pocket. I think to lock it in a conformation analogous to the Rossmann WIN compounds, I think, but I'm not sure. Good question.
Okay, so there is a Wei Yan who is raising his hand. So Wei, do you want?
All right.
Yeah.
Okay, so it's nice to see some friends. I just want to follow up with what Antonio commented. Also, the question, Jim. Oh, Martin, nice to see you.
Hey.
And so regarding this pronation depronation coupled conformal change, right? And also made comment a little bit. This is still unsolved problem. I would say it's an unsolved problem in the field of computational biophysics, 1D simulation. Although some people made some claim, right? Make some anecdotal claim, right? Published claims, right? And so they can solve it. They show agreement with experiment, with experimental, right? And so observations, either on PK or on conformal change observed from the actual structures. That's not an unsolved problem, right? Indeed, actually, it's very difficult. The reason it's difficult because the conformal change, the coupled, the time scale is very long, can be very long, particularly these low frequency motions. And so in order to particularly to get free energy value right, which corresponds to pKa, that's not a really easy thing to do. And then so particularly in the explicit solvent system, because the motion in the solvent system is more realistic. And so that's my comment, right? That's very challenging, right? And particularly for predictive, right? In your case, you need to have predictive, right? And it's different from benchmark work. People know the answer, trying to claim whether we can reproduce it. That's a different story.
Thank you. Yeah, interesting comment. I mean, of course, you're right. I mean, blind tests are what we should be doing, and what would be great would be if structural biologists, if we could produce anything interesting enough for structural biologists, or biophysicists, people who have the gene, can make mutations and do some biophysical tests on it. So that would be great.
Right. Just one more thing is because free energy surface for protein is, now we know more and more, even from experiments, it's very flat. It's not that sharp. It's pretty flat. So in that regard, actually, because pH change, a little bit pH change can be coupled with a lot of motions. But the motion is slow, right? Motion is slow.
So I have a question from the chat from Thomas Lauer. Do you think that a monovalent ion binding other than H+ is considered in any of the models?
For me, no specific ion binding other than H+, no. That's a very good point. I don't know of any specific, I've not come across spike structures where I've thought there's an ion binding that I should be worried about, and that's quite possible because it's cryo-EM, and it may be because it's cryo-EM and its resolution isn't there. I don't know. That's a good point.